Hello, thank you for joining us for our webinar. This month, we're pleased to again have an impressive panel of speakers that are here for round 2 of our questions about microbial myths. First up, we have Dr. John Wilson. Dr. Wilson really needs no introduction, as most people in the field have either talked with him for guidance or used one of his many documents that he's written. From 1978 until recently, Dr. Wilson worked as a highly respected research microbiologist at the R.S. Kerr Environmental Research Center in Ada, Oklahoma, where he led research dealing with biological and non-biological processes that destroy contaminants in groundwater. After retiring from the EPA, Dr. Wilson founded Scissortail Environmental Solutions. Then we also have another well-recognized speaker today, Dr. Frank Loeffler, who's the governor's chair at the University of Tennessee and Oak Ridge National Laboratory, with appointments in UT's Department of Microbiology, Department of Civil and Environmental Engineering, and Oak Ridge National Laboratory's Biosciences Division. Before moving to Tennessee, he was faculty in the School of Civil and Environmental Engineering and of Biology at Georgia Institute of Technology. Discoveries in the Loeffler Lab have contributed to the advancement of biodegradation and bioremediation, and he's contributed more than 80 peer-reviewed publications to the field. He also served on the ITRC Environmental Molecular Diagnostics team. And then finally, last but definitely not least, we have Dr. Kerry Sublette, who's also well recognized. He's a professor of chemical engineering and geosciences and Sarkis Professor of Environmental Engineering at the University of Tulsa. He has over 25 years of experience in a wide variety of bioengineering and soil remediation problems. Dr. Sublette has 13 patents and over 117 publications ranging from corrosion to brine spill remediation to ecosystem restoration. He chairs the annual International Petroleum Environmental Conference and also serves on the Oklahoma Board of Environmental Quality. So today we're really lucky to have these 3 speakers with such vast experience. And as many of you who have attended before know, the webinar today is being recorded and it'll be available on the Microbial Insights website within the next week. We'll send out a link to everyone and to all the registrants as soon as it's available. Now, we have a few questions that have already been submitted that we carried over from the previous webinar and from emails that have been submitted to us. So we're going to start with those questions. But if you have additional questions that you would like to do, then make sure you enter those into the GoToWebinar software so that we can get these experts to be able to answer them for you. Okay. So let's start with our first question here. So picking up where we left off last time, Frank, maybe you can start with this one. Can Dihalococoides be active in groundwater samples where the pH ranges in value between 5 and 6? And then the second part of that question is, what's the duration of Dihalococoides when it's exposed to a low pH that you don't see adverse effects? Would you expect that to be weeks, months, days? How does that work?
Okay, so we did some experiments over the last several years in a project funded by SURDUP, and we explored the effect of pH on Dihalococoides reductive dechlorination activity. And it's a little more complicated than we initially thought. So first of all, when you have defined conditions in the laboratory and you have a mixed culture containing Dihalococoides, you can manipulate the pH very precisely, and what we find is that if the pH drops below 6, reductive dechlorination activity is impaired. Specifically, the last dechlorination step, vinyl chloride to ethene, doesn't work so well. Now, if you take a culture, you grow at pH 7, and then you change the pH to 5.5, you will still see reductive dechlorination activity, but the organisms don't grow anymore. Dihalococoides doesn't grow at low pH, and what you're seeing is residual activity of those cells that grew at higher pH, and they can still do the process, but they don't grow. So it's not really a sustainable process at pH 5.5 or lower. The organisms don't grow at this low pH. So now we did also experiments where we actually added solids from different sites. and we adjusted the pH of the liquid phase to 5.5. And then we saw some surprising results. We monitored the pH in the liquid phase, and it was 5.5, and it stayed at 5.5. But in the presence of some soils, we saw reductive dechlorination activity and ethene formation. With other solid materials, we did not see any dechlorination and no ethene formation. So, and we always use the same culture to inoculate. So it's not a matter of the culture. The variable here was the soil from different locations that we added to these microcosms. My point here is that the properties of the solid phase probably influences microenvironments and that separate that the boundary layer on the solids from the bulk liquid phase, aqueous phase, in terms of pH. So even if the bulk phase pH that you measure is 5.5, it is possible that in the proximity to the solids, the Dihalococoides, possibly in biofilms, experience a different pH environment and can be active. So now, of course, the interesting questions, what are these properties of the solid material that enable that property I just described? And this is something we are exploring right now. And it's one factor that plays a role is the ion exchange capacity of the soil. But there may be other factors. And I have a student currently characterizing these soils so that we can then possibly predict under what conditions, under what properties does a soil, a solid material, have to have that you can expect Dihalococoides activity even though the bulk pH of the groundwater is 5.5 or maybe even lower. Okay, so just looking at the bulk, at the pH of the bulk groundwater does not necessarily tell you if reductive dechlorination activity is possible or not. So that's part of the question. I think there was another question you had, but now I forgot.
Oh, Frank, can I add to that just a moment?
Please.
Just in confirmation of everything you've been saying here, at a one complex site where we had a very large number of samples, around 280 samples at this complex site, one of the variables we looked at was the bulk pH and, of course, the DHC concentration. And interestingly, at this site, we found that we actually saw the highest DHC concentrations in the pH range from 5 to 6.5, and that was exactly our interpretation of those results as well, as that the bulk environment is not reflecting the microenvironment of the DHC.
Yeah, so I think this plays an important role. We are trying to get to the bottom of this so that we can, analyze site material in a way that we can predict how these dechlorinators will respond to low pH groundwater. Now remember the other part of the question. How resilient are these Dihalococoides to low pH exposure? So we did experiments without solids, just with liquid medium, and we exposed Dihalococoides cells that were grown at neutral pH to pH 5.5 for different periods of time. And we saw a correlation between exposure time and the activity we recovered after we adjusted the pH again to neutral. The longer you incubate Dihalococoides cells at low pH, the more cells die and you cannot recover activity. And if you keep it at low pH for 2 months or so, you will not recover any live Dihalococoides cells. So the exposure duration does play a role. Now, when we added solids with these, different solids have different properties to protect Dihalococoides. You can actually expose them to low pH, and even after extended period of time, you can still recover active dihalococoides from that solid material. So again, the solid phase can offer protection, but we do not fully understand what properties a solid phase has to have in order to offer this protection to dihalococoides. That's an open research question we're trying to find answers for.
All right, well, thank you, Frank. That was very informative. Okay, the next question that we have that was submitted is, are high sulfate concentrations toxic to the bacteria that assist in degrading chlorinated VOCs? And if so, what concentration of sulfate do you think would cause this toxicity? Carrie and John, do you guys want to take a stab at answering these questions?
Let me start. This is John Wilson. When Todd Wiedemeyer and I put together a scoring process to understand whether groundwater was a good habitat for dihalococoides, based on our empirical professional observations, we'd noticed that often when sulfate concentrations were high, we didn't see much evidence of biological reductive dechlorination. And I was aware of some thought and some work that was done by Chuck Newell at Groundwater Services, Inc., who reasoned that the sulfate-reducing bacteria have a higher affinity for hydrogen than many other anaerobic bacteria. And if sulfate is available, the sulfate-reducing bacteria would proliferate and pull the concentration of molecular hydrogen down so low that other bugs like dihalococoides couldn't function. And so it's not a toxicity, and it's not even an inhibition. It's basically stealing a requisite substrate, outcompeting other organisms. So it's not a toxic interaction if it occurs. It's just simply that the sulfate-reducing bacteria, because of an advantage in the thermodynamic, gives free energy, just can do a better job of poisoning hydrogen concentrations at low concentrations. But it turns out that that was very plausible. It was reasoning from first principles, but people have actually in recent years had a chance to actually examine that in some detail, and the reasoning is not very useful. There is an effect, but it's not black and white, on and off, live or die. It's sort of, well, it slows them down a little bit. And Dr. Sublette actually has a data set that he's been working on from field sites that gives better resolution of what the effects of sulfate concentrations truly are on the activity of dihalococoides. And so, Carrie, it's your turn to pick up the story.
Sure. What we found at this one particular site, it was a very complex site with multiple chlorinated hydrocarbons and many sampling wells. We looked at, among other things, looked at the DHC concentration and sulfate concentration and to see how they correlated at the site. And there was a negative correlation between the log of the DHC concentration and sulfate concentration over a range of about 0 to 700 milligrams per liter. But there was no step change in the DHC count with sulfate. It was a gradual decrease as the sulfate concentration increased. Now, we could possibly determine or define a threshold value based upon where the DHC concentration dropped below what is generally considered to be a useful concentration, that is 10 to the 4th cells per mil. And it was at about 50 milligrams per liter sulfate is where we did drop below that particular threshold. But over that entire range, out to 700 milligrams per liter, it was a gradual decrease in DHC activity.
At other sites, I have seen ethene production in waters that contained 100 milligrams per liter of sulfate. So the short answer is that yes, it probably slows it down, but as Dr. Sublette said, it's not a step change. It doesn't turn it off altogether.
I can add maybe a little information here. So we did experiments exploring if sulfate or the products of sulfate reduction, hydrogen sulfide, HS minus, would influence the helicocoides activity. And we did not see a pronounced effect of sulfate in pure cultures. And we did not really see a pronounced effect of HS minus, so the product of sulfate reduction, sulfide. It also did not significantly inhibit the helicocoides activity. So that was done in pure cultures. If you do this in mixed cultures, it may look a little different, what was just discussed. It is possible that sulfate-reducing bacteria that are ubiquitous will consume electron donor and couple electron donor consumption to sulfate reduction. And so then you may have a competition situation where the helicocoides has to compete with other bacteria for hydrogen. And that may have an effect on activity.
So I'd like to add something to that. There are techniques to measure molecular hydrogen concentrations, and it's very easy to measure the concentrations of acetate. And that's what the helicocoides is looking at and cares about and influences its activity. So if there's plenty of acetate and hydrogen, it doesn't matter how much or how little sulfate is in the water.
Okay. Very good answer. Okay, let's go ahead and move to the next question. Frank, I think this is a good one for you to start with. What's the detection frequency of the helicocoides in functional genes? How often are you seeing them at the sites? And if the detection limit is low enough, would you expect that we would find the helicocoides everywhere?
Okay, let me just add one thought to the sulfate discussion that I forgot to say something about. So there was an observation published recently that organisms can actually consume ethene under sulfate-reducing conditions. So they can couple ethene oxidation to sulfate reduction. So when you have a lot of sulfate present, it is quite possible that you do not detect ethene because it is consumed under sulfate-reducing conditions. So you have to be a little cautious about, not detecting ethene in the presence of sulfate does not mean that reductive dechlorination is not working. It may simply mean that the product of reductive dechlorination, ethene, is consumed. So that's one point I wanted to make about the sulfate discussion. So now about the question, what are the detection limits for the helicocoides? So the most sensitive technique we can apply is quantitative PCR that targets specific biomarker genes of the helicocoides. And this technique has a relatively low detection limit. And a little higher than the detection limit is the limit of quantification, where we can actually tell how many of these biomarker genes representing the helicocoides are present. And Dora, you may know the numbers better than I do, but you need about 100 dihelicocoides cells to detect them and be able to tell how many are in your sample. So it's a very low, relatively low detection limit. So we can detect the helicocoides when they are in low abundance, present in the groundwater or in a soil matrix. Now, in order to link the abundance to reductive dechlorination activity, the general assumption is that you need at least 10 to the 4 per mil, so about 10 to the 7 per liter, to see robust reductive dechlorination activity. So the technique to detect is much more sensitive than the abundance you would like to see in order to have good reductive dechlorination activity and degradation performance. So now, Dora, what was the other part of the question?
If the detection limit is low, do you expect that we would see dihelicocoides everywhere?
Oh, so this is an interesting question. So let me try to answer that from a different perspective. So we know enough about the Helicoccoides biology that we understand what the organism needs in order to proliferate, to grow. It needs an anoxic environment because it cannot deal with oxygen. We also know that it requires hydrogen as electron donor, so it needs hydrogen. It also needs chlorinoid because that's a critical cofactor for reductive dechlorination, and it can't make it. So it must live together with other organisms that supply this growth factor. It is sensitive to carbon monoxide, and it actually, in its metabolism, the Helicoccoides produce small amounts of carbon monoxide. So it must grow with partner populations that consume carbon monoxide. Then the Helicoccoides requires a halogenated compound, a chlorinated solvent, for example, as electron acceptor. Without that, it can't grow. So now you have to put this all together, then you can better define the environment where the Helicoccoides can occur. And if you think about this more deeply, even in oxic soils, you will find anoxic microniches. And people have demonstrated that in those microniches, microenvironments, you can find the Helicoccoides. So my answer to this question is, you will find the Helicoccoides probably in most environments where you can expect anoxic conditions, even in microenvironments, where you have some hydrogen flux and where you have some chlorinated or halogenated organic compounds. And here, don't think about anthropogenic contaminants. Think more about naturally produced halogenated organic material. Even PCE, even vinyl chloride, can be produced by natural processes in soil or in other environments. So these compounds are present in very low concentrations. And those are probably the reasons why the Helicoccoides can make a living in environments that are not contaminated. So the more we look, the more often we find the Helicoccoides in environments that are not contaminated by human activities. So my prediction is the Helicoccoides is in most places where we can expect anoxic conditions to occur.
And I can add just a little bit to that, that Frank was saying is when we looked at more than 12,000 samples that we've received for the Helicoccoides analysis, now granted these are typically from contaminated sites, we see that the Helicoccoides is detected around 70% of the time. And the other, I guess, caution on that is saying that it's detected, as Frank mentioned, we have very low detection limits. So it doesn't mean that the Helicoccoides is typically at that 10 to the 4 cells per mil that you would want to see for a more robust reductive dechlorination to be occurring. But from our experience from those more than 12,000 samples, it's around 70% detection. Okay, so let's move to our next question and take this one step further. One of the questions that was submitted last time is, have any of the speakers experienced sites where no degrading bacteria were present? I think this is something that you can probably all contribute to. Terry, do you want to start with this one?
Well, sure. I've had no such experience. Any sample of soil or groundwater or surface water that I've ever looked at contains copious numbers of organisms, including various types of degraders. I'm more often than not looking at hydrocarbon degraders, and as we all know, they're basically everywhere. I've even looked at groundwaters where I thought the conditions were such that the numbers might be severely depressed. For example, looking at groundwaters that had been impacted by salts, chloride concentrations in excess of 15,000 milligram per liter in the groundwater, and still seeing very high numbers of organisms. I've looked at soils that have been impacted by produced water spills, and here in Oklahoma, produced water from oil and gas production can have TDSs of in excess of 300,000 milligram per liter directly onto those soils, and you sample those soils and you find, yes, there's been an impact, but I've never seen more than a 50% reduction in the soil microbial population. So you know, the bottom line is I haven't seen one yet. The only data that I have ever seen in which it was purported that a particular soil environment was sterile and that they could not find any degraders or any organisms at all was actually a site in Europe where they had been using electrical resistance heating in the subsurface, and the soil had been heated to a very, very high temperature. But that's the only case I have ever seen.
Dr. Wilson, do you want to add additional experience to this?
Well, actually, early in my career, I worked with Herb Ward on a site that basically it was his work. He had the observation that there was no biodegradation activity, and he added active cultures and in material and microcosms and still got no biodegradation. And so, building on what Dr. Sublette just told you, the real question is whether there's activity, not whether there's the presence of capable organisms. And I think that's a better way to frame the question. There are sites where conditions preclude significant rates of biodegradation, whether organisms are present or not. And so what I've done really is reframed the question from the participant. As Dr. Sublette said, you have to go to quite a bit of trouble to find material in the earth that's sterile. People that have looked deep in the earth to try to find regions of our planet that don't have biology, anytime they look carefully, even kilometers deep, they'll find organisms. So I really think that the absence of degrading bacteria does not explain the absence of degradation. If you don't have degradation, the question is what's precluding those bacteria from being active?
I think this is a very important observation, that the environment must allow the degraders to be active. For example, if you have the Helicoccoides present and they can degrade the contaminant of concern, but the hydrogen flux is so low that they don't have the electron donor available that they need, you will not see degradation. So the environment is not appropriate. And now you can influence the environment by doing biostimulation. Now in some cases there are no degrading bacteria present. Bacteria are there, but not the ones that have the right pathway, the right genes and enzyme systems to degrade the contaminant efficiently. Maybe these organisms are present, but in such low abundance that you can't detect them initially, and then it takes some time before they increase in abundance and you see activity. The reasons why certain pathways are not represented in an environment where the contaminant is present, it's not really clear. But the bigger picture question is, can we answer how long it will take for the organisms to acquire the capabilities, to acquire the genes and the pathway to degrade the contaminant? Can we develop enough understanding of the biology to predict how long will it take for a pathway to emerge? Can we predict how long it will take a Helicoccoides cell to pick up a gene that efficiently dechlorinates vinyl chloride? That would be very helpful to develop predictive understanding. In the short term, we can use bioaugmentation cultures and introduce organisms with these genes and pathway capabilities to sites where we believe that the right organisms with the right genes and the right pathways are not present. that would be my addition to what was discussed by the other participants.
Thank you, Mike. Dr. Hyman, are you on the call now as well? We had some technical difficulties, but I think he's been able to join. Okay, thanks. So Mike Hyman is a professor at the University or North Carolina State University, and he, I think, can add a lot to this as he has more than 30 years of experience in biodegradation of different environmental pollutants, particularly a lot of work with MTBE that's been very interesting. So Mike, do you want to add anything to the discussion on this?
Well, no, I think everybody did a really comprehensive job on it. The one thing that you might want to think about is that you know, a lot of organisms, when they're stressed, well, not a lot, but a significant portion of microorganisms have the ability to generate what are effectively spores and go into this sort of quiescent, non-active state. So certainly some hydrocarbon oxidizing bacteria can do that. So again, you know, you can have no biodegradation occurring, but the organisms are still present. They might just not be in a particularly happy or healthy state at the time.
Okay, great. Thank you. Okay, so the next... I'm sorry.
Before we move on, let me add one more detail to my response. I've done a lot of microcosm studies where I take samples of material from sites that look like something's happening and see if I can reproduce it in little glass bottles in the laboratory. One thing that I noticed early on is the chance of reproducing the field scale effect increased with the size of the microcosm. So if I started with a little sample like a gram, I often had a lot of variability in the microcosm response in replicates. It grew up in some microcosms and not in others. And as I increased the size of the sample that was queried, the closer it came to mimicking the field scale system. So yes, if you take a tiny sample of an environment, there's a possibility that you could miss the bugs that are there in the large field scale system. So part of it might actually be a sampling artifact. Yes, it is possible to take a tiny sample and miss a population.
That's a good point. Okay, our next question goes a little bit into petroleum degradation. So maybe Dr. Hyman, if you want to start with this, then Carrie can finish and elaborate more. But what parameters do you suggest that we really think about and be aware of in terms of cell population and activity for petroleum degraders?
Well, from my perspective, I work on you know, individual types of microorganisms rather than populations as a whole. So my answer is going to be kind of dictated by the fact that petroleum contains this enormous range of different compounds, all of which, well, many of which can be degraded through different mechanisms and by different microorganisms. So you know, from my perspective, it's you know, your interest or your focus should be on what particular portion of the petroleum mixture is that you're trying to biodegrade. So, for example, you know, for gasoline-related issues, one of the primary interests is the most toxic compounds, which are things such as benzene and some of the other aromatic compounds. I'm not quite sure what else I could add to that. What parameters should we be aware of in terms of cell population activity for petroleum degraders? You know, the environmental conditions, we've already talked about that to some degree. You know, a range of reasonable pH. A lot of petroleum biodegradation occurs very rapidly under aerobic conditions. You get a lot of anaerobic degradation, typically slower. Yeah, I mean, it's an incredibly broad question. I think perhaps maybe Kerry could give you a better perspective from the population size, but from the individual organism side, I think I've covered most of the points there.
Well, from the population side, I've done a lot of work both in groundwater and soils, looking at hydrocarbon degradation, and certainly one of the things I think most everybody knows at this stage is that petroleum degraders are ubiquitous. They're just everywhere. I've not found an environment where I cannot find at least some level of hydrocarbon degraders. So when DNA is sampled and we look for catabolic functional genes, whether they be aerobic genes or anaerobic genes, we almost always find them, which kind of makes it difficult to document any stimulation that perhaps you're trying to enhance in terms of hydrocarbon biodegradation. But what we have found is that if you find these same catabolic genes in the RNA, it's much more instructive, and we know that the genes then are being transcribed and hydrocarbon degradation is likely going on. And then once we verify the genes, Dr. Hyman was mentioning the geochemistry, looking at the geochemistry and seeing if the geochemistry changes that we can document are consistent with the metabolic processes that we think that we're seeing. I'll give you one little quick case study. We were doing a study with an in situ bioreactor in an aquifer impacted by fuel oil. And we had been using biotraps to monitor populations of hydrocarbon degraders. And if initially, before we started using any amendments or installing the bioreactor, if we looked at catabolic genes in DNA, they were certainly there, as we might expect. But if we looked for them in RNA, then they were in very low concentrations or even non-detect. But after we initiated the operation of the in situ bioreactor, the same genes that were showing up in DNA now start showing up in RNA, providing direct evidence that we've stimulated hydrocarbon degradation. So I would certainly advise anyone that wants to document in situ documentation of hydrocarbon degradation to think in those directions. The DNA will certainly tell you they're there, but as was pointed out earlier, they can be there, but they're not necessarily active. You need an indicator of activity, and that indicator of activity, in my mind, would be finding those catabolic genes being expressed in RNA.
Actually, got a couple of other things that I can probably add to that. As Dora pointed out, some of my experiences in the area of MTBE, one of the poster childs for MTB biodegradation is Methylibium strain, Methylibium petrophilium PM1. And when you study that organism in pure culture, what you can find out is that not only does it degrade MTBE, but it also has the ability to grow on and utilize aromatic compounds. So one thing that you could potentially be blindsided by the fact is that you're looking for MTB biodegradation, you find PM1 or something similar to PM1, but in fact it's not necessarily degrading MTBE. It could well just be quite happily degrading a much more readily biodegradable compound such as benzene or toluene or something like that.
In fact, if I could add to that just a little bit, Mike, I did a survey at a refinery site where there had been no MTBE utilization or certainly no releases into the groundwater. And we did a statistical study looking at concentrations of various genes, including PM1. And we found that there was a statistically valid correlation between concentration of PM1 that we detected and toluene concentrations in the groundwater.
Right. So, yeah, I mean, a lot of hydrocarbon oxidizing bacteria, which is what we're primarily thinking about when you deal with petroleum degraders, often have more than one capability encoded in their genome. So they have many metabolic capabilities, and it's just as a general rule, those types of organisms are going to degrade the more readily assimilable compound or the most abundant compound in preference to something that may be regulated at a much lower concentration than something that you're looking at. The second point that I wanted to point out was that when you have complicated mixtures of compounds such as you would do in a petroleum contaminated environment, whether it be diesel or crude oil, whether it be gasoline, what you find is that there is a lot of catabolic, in other words, degradative capability, tends to one enzyme can actually degrade multiple different substrates. And so, for example, you could have an organism that's growing on a straight chain alkane, something like pentane, hexane, something that's abundant in gasoline, and at the same time it has the expression of that enzyme enables that organism to degrade other compounds, but not actually utilize the organism that's catalyzing this reaction doesn't actually utilize the byproducts of that reaction. And again, you know, if you were looking at, for example, degrading pentane, and actually the compound that you're particularly interested in is benzene, it could well be that benzene degradation is occurring, but it's not occurring through the activity of organisms that are necessarily growing on the benzene. That transformation could be catalyzed by organisms growing on the straight chain alkanes. And the more complex the mixture, the more likely that type of process, I think, is likely to occur. And so generally that's sort of referred to as a co-metabolic transformation. And that makes it very difficult to unequivocally identify which particular organism or groups of organisms within a population are actually responsible for a particular activity.
Dora, could I add a little more?
Sure.
To this question?
That'd be great.
In many petroleum sites, the wells are screened across the water table, and as a result, you have water contributed to the well from a zone that has oxygen and hardly any hydrocarbon. Water contributed from zones that have hydrocarbon and essentially no oxygen, and then a little bit of water that's contributed from that interface that has both, and that's probably the water that's producing the organisms. And so we take this mixed water sample and send it off to Microbial Insights and find that there are a lot of organisms in it, but that does not mean that the organisms in the well water were in the water that contained the contamination. And so you really can't extrapolate an implied activity across that entire screened interval. The organisms most likely are being contributed from a different flow path than the flow path that contributed to the vast bulk of the hydrocarbon contamination. So that doesn't mean you don't disregard it. There's a lot of information in the gene information. Moving back to Dr. Sublet's point that if you actually give the bug something to breathe, then you see RNA. The presence of those organisms indicate that if you do decide to go with an aerobic treatment system that provides oxygen, the bugs are ready, willing, and able to do the job for you as soon as you remove that geochemical constraint on their activity and allow them to metabolize the hydrocarbon.
John, can I follow up with that on a comment regarding the complexity of the groundwater monitoring well and the different environments, vertical environments? One way we have attacked that problem in the past is by using biotraps that are suspended by a float from the surface of the groundwater, so that we can actually suspend the biotraps at whatever depth interval we want, and so we can get a picture of how that microbial population changes with depth.
So the point of that is that if you look at the system in 3 dimensions, then it tends to make a lot more sense. Can I move on to the...
Okay. The next question.
I could move on to the question about MTBE.
Okay. Yeah, that's the next question that we have, is that are there effective in situ approaches to the remediation of MTBE?
And the answer is yes. And there's a whole book about it. The title of the book is MTBE Remediation Handbook. It was edited by Ellen Moyer and Paul Kostecki. Let's see, when was it published? It's been a while. 2003. But there's lots of documentation that if you can get oxygen into MTBE contaminated groundwater, and give it a few months, PM1 will take care of the MTBE. So aerobic bioremediation of MTBE in situ is effective when people do it right. And I'm not going to add any more to it unless somebody, one of the other panelists, has some additional insight or depth to comment on it.
Well, I would certainly agree with you, John. Adding oxygen into a contaminated environment would be the number one go-to approach. You can certainly biodegrade MTBE anaerobically, but it tends to be certainly the more reducing conditions, the less complete that oxidation or that degradation appears to be. And so you may end up with the production of the byproduct tertiary butyl alcohol, which then represents, its own set of challenges. I would draw a distinction between sites or environments that have MTBE by itself, as opposed to MTBE in as part of a mixture with unoxidized other gasoline components. And again, that comes back to what I was discussing in the previous answer. There are certainly organisms that can grow on and utilize MTBE as the sole carbon and energy source. And those, for example, are strains like methylibium PM1. But it's also very, very clear that a lot of other organisms can partially degrade MTBE. And the most frequently encountered ones are the ones that grow on primarily the alkane components of gasoline, so straight chain alkanes, branched alkanes. And those organisms have the ability to co-metabolically degrade MTBE. So in fact, they're growing on the alkane, but then they're fortuitously degrading that MTBE. And so if you were to oxygenate an environment that contained MTBE by itself, which given its transport properties you can encounter, you might expect and might be advised to look for the presence of PM1 and its characteristic genes. I think the higher the level of contamination or co-contamination with unoxidized gasoline components, the less reliable that metric becomes, and the more likely that co-metabolic degradation, again, under aerobic conditions, is contributing to that degradation process.
Thank you, Mike. That's a good point. Okay, and the next question that we have kind of moves off topic a little bit, and I think a lot of our panelists have experience with this, and maybe Dr. Wilson, you want to start. But how significant do you consider abiotic degradation mechanisms to be in the environment? And do you have any information that's available, particularly on iron oxides?
Well, they are important for natural abiotic degradation of alkanes in particular specific groundwater environments. If you have an unconsolidated sand aquifer that is comprised of material that's sourced from weathering of granite or other metamorphic rock, or if it has a significant component like certain categories of glacial outwash sands, you can have enough magnetite naturally occurring as one of the components of the sediment to produce rates of degradation of the chlorinated organics, alkenes, in the order of 10% or so a year. Now, if you've got a lot of time and a long fetch for your plume, but before it reaches a receptor, these relatively slow rates can provide significant protection. So it's significant if a slow rate of degradation is useful, and it's significant if you have an aquifer that has magnetite in it. The other fairly well understood category is material that has iron monosulfide, mechanoide. formed usually from biological sulfate reduction producing hydrogen sulfide that reacts with natural iron minerals in the aquifer matrix. This can be significant too. The rate constants often are much faster, but it's always coupled to biology. You have to have something driving the sulfate reduction. And very commonly, if you have enough carbon substrate for sulfate reducers, you're also making enough hydrogen for dihalococoides. And if you look at the complete system, the contribution of the bacteria is more than the contribution of the abiotic mechanism. The abiotic mechanism may contribute maybe half, maybe 10% to the total activity, but it's not the primary story. We're starting to learn that there are, in fact, other naturally occurring minerals that can contribute to this, particularly in anaerobic environments. It's not to the point now where we can measure something, though, and associate it with a rate constant, but certainly glauconitic sands, green sands, have this capacity. And there are certain other iron-2 clay-like minerals and clay-like things that are associated with diorites. Things are associated with limestone-like minerals that are found in metamorphic rock in the East Coast. We're just now learning about how that works. But the significance of the abiotic degradation processes is a niche. It's not a uniform and universal answer to any problem. It's kind of hard to manipulate. So if you're lucky that you have enough of it that it's useful at your application, well, then it is significant. But most of the time, probably 80% of the time, it doesn't solve your problem. In terms of information available on the iron oxides, I helped edit an EPA report on methods for analysis of materials and minerals that contribute to abiotic degradation. And I encouraged Microbial Insights to actually offer an analysis for magnetic susceptibility, which is a very good surrogate for the presence of magnetite in aquifer sediments. So these analyses now are commercially available through Microbial Insights.
I can add a little bit to the magnetite significance of magnetite for abiotic degradation. And so it's well known that magnetite can be a very good mediator for degradation of carbon tetrachloride. So carbon tet is not really degraded very well by the microbiology because it's a very toxic compound to microbes. But carbon tet is not stable in the presence of magnetite. So Dr. Wilson mentioned magnetite that is coming from rocks. But there's another possibility how magnetite is generated in anoxic environments like aquifers. So ion-reducing bacteria, organisms that reduce goethite or ferric ion, solid materials, can generate magnetite. There's also evidence that ion-oxidizing bacteria, so they oxidize Fe2+ to Fe3, ferrous ion to ferric ion, they can also contribute under certain geochemical conditions to magnetite formation. So there are other sources for magnetite. And dependent on the geochemistry of your aquifer, it is possible to generate additional magnetite based on the microbiology that is present and active. And so, for example, in carbon tet plumes, the formation of magnetite can be a major contributor to carbon tet degradation mediated abiotically by magnetite. So magnetite appears important, and what Dr. Wilson just mentioned is that it's relatively easy to measure and also quantify. So then you have an idea about the amount of magnetite that is associated with the aquifer material.
I'll throw one very, very odd little curio into that answer as well. It turns out that there are and I think they're primarily aquatic organisms as opposed to, for example, organisms that you'd find in the subsurface. But some bacteria have the capability of making what are called magnetosomes, which are small crystalline structures of iron oxide. And I don't know whether anybody's examined whether these organisms have the ability to biodegrade anything, but it's sort of listening to this conversation, it's a thought that might be worth following up.
So this is correct. So they are magnetotactic bacteria, ubiquitous pretty much, and they generate magnetite intracellularly for, well, they can orient themselves in the magnetic field. That may have an advantage. It's not really clear what it is, but they can do that. And they obviously have then magnetite produced. When they lyse, the magnetite becomes available. It's nanoparticulate magnetite that is active. However, the amount of magnetite these organisms contribute to the overall amount in an aquifer that is already present from the weathering of rocks and then from ion reduction or ferrous ion oxidation is probably marginal. So they can contribute, but their relative contribution is probably not significant.
I'd like to add something to what Dr. Loeffler said. If I were going out to try to find an aquifer where biogenic magnetite were important, I'd look for a source that had organic matter along with the carbon tetrachloride. For example, where there was co-disposal of chemical manufacturing waste and conventional municipal solid waste in an uncontrolled landfill, something like that. So that I have carbon that can be degraded under anaerobic conditions co-occurring with the chlorinated solvent. Then I would look for groundwater that didn't have any or much sulfate in it, so that iron reduction is the dominant sink for electrons produced during the fermentation of the organic matter. In those conditions, I think there's a very real good chance that magnetite would be produced and accumulate. And as Dr. Loeffler mentioned, and I should have gone into in some detail, that biogenic magnetite made by the bugs, is much more effective than the magnetite that weathers out of the granite, maybe 10 to 100 times more effective. And so if it is being formed, it can in fact really control the distribution and fate of certain classes of chlorinated organic solvents.
Okay. Moving on to our next question. This is one that was submitted. It says a comment, thank you for arranging these webinars and letting people from all over the world participate. It helps very much and I really appreciate it. So I thought you panelists would really enjoy hearing that. Their question is there any experience in adding electron donor into coarse sand aquifers? Is it more a matter of quantity concerning the higher groundwater flux, or what type of electron donor has been chosen, soluble versus slow release type compounds?
I'll start so the other panelists have a chance to organize their thoughts. And so with the caveat that mine are not organized, the problem with aquifers that have a high seepage velocity is that your amendment tends to get flushed away. Your goal, in fact, is to try to mix your amendment with the contaminated water. It's pretty well established the best way to do that is to get your amendment to somehow stick around in the area where you inject it. And then as the groundwater continues to flow past that injection zone, then it picks up a little bit of the amendment, enough to do the job, and moves away for activity. And so the best way to mix the amendment into the flow of the aquifer is for the amendment to be relatively stable in space while the groundwater moves into the amended zone, gets the benefit of the amendment, and then gets treatment. Now that's very hard to do with very soluble amendments. They tend to flush away as well. They don't entirely flush away, but they have that strong tendency not to stay put. That's why for many of these high flow aquifers, the best approach is to add the amendment as an oil, a veg oil, often emulsified, but with surfactants that control its tendency to move a little bit and then change its properties and sorb onto particles. It doesn't mean that you can't treat a high flow aquifer with entirely soluble materials. It's just that it's a lot more of a challenge from an engineering point of view. But your goal really is, to reemphasize what I said, is to mix your amendment with the flowing water. And so if you can find some way to do that, perhaps with recirculating wells in a dipole or something like that, you could also have success with soluble amendments. But if you're not, if you're just trying to simply pump your amendment into the aquifer to treat it, then something that tends to stay put is going to have more efficacy.
Okay, does anyone else have a comment on that?
I mean, I have just a general statement. So what you want to do is whatever you add to an aquifer, the goal is that you increase the flux of hydrogen, because this is often a limitation for these organisms that perform reductive dechlorination. So now you can use different substrates, electron donors you add, and typically these are fermentation reactions, and you can generate a lot of hydrogen in a short period of time, or you can generate a low flux of hydrogen over an extended period of time. If you have a lot of hydrogen produced initially, it's very likely that the majority of this hydrogen produced is consumed in other microbial processes. So this may not be an ideal thing to do, because a lot more of the electrons you're supplying with the substrate addition are directed to other microbial processes that don't benefit your goal of what would be contaminant degradation. The other issue, I think, what should be considered and should maybe get a little bit more attention are secondary effects of substrate addition. So there are other effects that can happen following substrate amendment. I just mentioned fermentation reactions occur, and those typically are associated with the liberation of organic acids. Then in addition, you stimulate reductive dechlorination and you generate more acid because you basically release hydrochloric acid from reductive dechlorination reactions. Then the pH changes, and that may then trigger secondary effects that ultimately lead to degradation of the aquifer. The secondary effect can contribute to degradation of the quality of the groundwater. And these are things you need to consider up front, because it's not just removing the contaminant that should be the goal. You also need to keep in mind the overall health of the aquifer. And so those ideas should contribute to deciding on the most appropriate amendment to achieve the goal, what would be contaminant degradation, without impacting the aquifer in any other way. Does that make some sense, what I just said? I think I was not as organized as I wanted to be.
No, I think it makes sense, and it's really an important consideration that we don't take into account often enough.
Right, so another issue that is related to this is the amount of methane you're producing. Right, so methanogenesis is a process that probably will occur in most aquifers, and you can determine the amount of methane produced by the type of substrate you're adding, and also by the quantity of substrate you're adding. And producing a lot of methane is probably not in the interest of the person in charge of the remediation effort, and it's also a greenhouse gas. If it's released, it's not a good thing. It's a negative secondary impact of amendments. And those issues should receive greater attention so that we can implement what I would call precision bioremediation, where you specifically target your contaminants without inducing other negative secondary impacts of the treatment.
Dora, I didn't have anything to add to that last question, but seeing as you still have this slide up about abiotic degradation mechanisms, and we were talking about biogenic metal oxides, the one other one that I'm aware of is that there are a number of bacteria and fungi and other organisms that have the ability to oxidize manganese. And they make manganese oxides. And manganese oxides are extremely potent oxidizing agents. And these manganese oxides tend to accumulate on the extracellular surface of the bacteria. And there's certainly interest and demonstrated capability that these manganese oxides can act as catalysts for the degradation of compounds that are present at very low concentrations. And in fact, that's one of the drivers, or at least one of the better ideas that I've seen out there about how that type of process could be utilized, is for the degradation of some of these very, very bioactive compounds that people are concerned about these days, these personal care products and pharmaceuticals, where they seem to flush their way right through wastewater treatment plants, primarily because they're present at such low concentrations. And yet you can then have microbial systems where the organisms that are generating these manganese oxides can then oxidize those compounds and detoxify them. I don't know whether folks have looked particularly as far as the type of contaminants that we're interested in or have been discussing today, but again, you know, manganese oxides would be included in that group of reactive metals that biology can create.
All right, thank you. Unfortunately, we've gone over time. We still have a big list of questions that have come in from the audience, so perhaps we can convince our speakers to either help us answer those or do a round 3 of this in a few months. But thank you very much, Dr. Hyman, Dr. Wilson, Dr. Loeffler, and Dr. Sublette for being with us today. This is, again, really informative, and I think our audience has enjoyed it. And for those of you out there in the audience, we appreciate you attending. And if you have additional questions that you think of over the coming weeks, please email them to info@microbe.com, and we'll get with the speakers and figure out how we'll do this moving forward to get some more answers for you. We appreciate your attendance and the questions that you provided. And again, panelists, we really appreciate you answering these and providing your expertise for the environmental community. We really appreciate it. All right, everyone have a good day. Thank you very much.